Hello and welcome. Today, we're diving into a piece of technology that is, and I'm not exaggerating here, a total game changer for scientific research. It could completely rewrite the rules for how we study our biggest health challenges.
So let's just start with a really big question. What if the scientific data we gathered in a lab today could be perfectly matched up with data collected, say, 10, maybe even 20 years from now, from any lab anywhere on the planet? Think about what that would mean.
To really get why that's such a huge deal, we first have to talk about a massive problem in science today. Let's call it the data dilemma.
Okay, think about this for a second. To really understand chronic diseases, stuff like Alzheimer's or diabetes, you need to track biological changes in things like our blood and urine over a whole lifetime. But here's the problem. Our scientific instruments are always getting upgraded. The data from a machine built in 2024 often can't be compared to data from a machine built in 2034. It's like they're speaking two different languages.
This creates these things called data silos. It's a huge headache. A lab in Germany might have this incredible, valuable data set, but they can't just merge it with a similar one from a lab in the UK that's using a slightly newer machine. All of this amazing information is trapped, isolated. It makes those huge, long-term studies incredibly difficult, if not impossible.
But what if there was a way to measure something so fundamental, so absolute, that it just doesn't matter what machine you use to measure it? Well, that's where this idea of a molecular fingerprint comes into play. And that brings us to a technology with a, let's be honest, very long name: Fourier Transform Ion Cyclotron Resonance Mass Spectrometry. We'll just call it FTICR-MS. At its core, this amazing machine traps charged molecules in a super strong magnetic field and then measures their unique wobble. And that wobble, or frequency, is directly tied to their exact mass, giving us an incredibly precise measurement. Now this is the T. This is the whole shebang right here. FTICR-MS measures a molecule's exact mass, which is an absolute physical constant. It doesn't change, ever. It's like a permanent, unforgeable fingerprint for every single molecule, a fingerprint that any FTICR-MS machine, anywhere in the world at any point in time, can read. And when I say precise, I mean it. Look at this number. This represents the average error from a major study. We are talking about an error of less than 1 part per million. It's almost unbelievably accurate. And it's that level of precision that makes the data so reliable, so trustworthy, and potentially timeless.
So the theory sounds fantastic, right? But does it actually work in the real world? Well, researchers decided to put this idea through its paces. First up, can two totally different generations of machines actually produce the same results?
For the first test, they pitted an older machine in Germany against a much newer one in the UK. We're talking about a technology gap of nearly 2 decades between these instruments. And to make it really tough, they fed both of them complex pet food samples, a really messy mix of all sorts of molecules. And would you look at that? The results were incredible. After they carefully tweaked the settings to make sure both machines were on the same page, the molecular fingerprints they got were practically overlapping. The performance was similar, and the overall chemical profiles were directly comparable. That is a huge first win. Let's dig into the numbers a bit. If you look at this chart, you can see that key performance metrics, like how clearly the signal stands out from all the background noise, were amazingly similar for both the old and the new machine. This just shows that even with all their differences, their performance could be perfectly harmonized. And it's the exact same story here. The number of different molecules they could detect, and, most importantly, that incredibly tiny mass error we saw earlier, it's all in the same ballpark. This is solid proof that with the right setup, the data from these two very different instruments is basically interchangeable. But this, right here, is the real mic drop moment. This is a statistical plot, and what it's showing us is that the samples are grouping together based on their actual chemistry. You see the wet food is over here and the dry food is over there. But here's the crucial part. They are not grouping based on which machine analyzed them. The machine became irrelevant. The only thing that mattered was the science. Okay, so it works for 2 instruments. That's great. But can you scale it up, like globally? Could you get labs all over the world to produce the same comparable data? That was the second and much bigger challenge. To figure this out, they organized what's called a ring trial. They sent identical samples of human plasma to 17 different labs across 4 continents. And as you can see from the chart on the right, yeah, getting everyone's data to line up perfectly was definitely a challenge. It really showed how differently these powerful instruments are used from lab to lab.
But, and this is a big but, a core group of 5 of those labs, using different instruments with different specialties, managed to produce results that were highly comparable. This was a massive breakthrough. It proved that with standardized methods, creating a global network of data that can all be integrated isn't just a fantasy. It's actually possible.
All right, so we've established comparability across different machines and across different labs. But what about the final frontier, the ultimate test? Comparability across time itself.
You know, as this quote from a 2025 paper points out, before this research, no one had ever really shown that you could take data collected over many years and truly merge it all into one single, continuous river of information. And that's exactly what these researchers did. This timeline shows an absolutely heroic effort. Over 6 years, they analyzed hundreds of complex samples from large groups of patients, all in 32 separate batches. And because FTICR-MS was capturing that permanent molecular fingerprint every single time, they could stitch all of that data together into one enormous, seamless, and ever-growing database. So, we've walked through the problem, the tech, and the proof. But let's get to the bottom line. What's the real payoff here? Why does creating this kind of timeless data actually matter so much?
Well, the implications are just staggering. Just imagine being able to track the molecular story of thousands of people over their entire lives. We could potentially spot the chemical signs of disease years, even decades before any symptoms show up. This technology makes those kinds of long-term studies a reality. It means researchers around the world can finally pool their data together, creating these massive databases for discovery. And it ensures that the billions we invest in research today won't become obsolete. Its value will just keep growing for generations. Ultimately, this shifts us from taking isolated snapshots of biology to watching the entire movie. With data that truly never expires, what mysteries that take a lifetime to unfold will we finally be able to solve? From chronic disease to the health of our planet, the future of research is here, and it looks like it's timeless.